Hi. So there's been many studies looking at coral bleaching using bulk RNA-seq methods. And while this is great for looking at whole organismal responses during thermal stress, it lacks the resolution to ask more targeted questions about the cellular processes that are happening during bleaching. So my project is using single-cell RNA sequencing to understand which cell types are most affected during heat stress in corals, and then can we actually mechanistically understand coral bleaching pathways at a cellular level. So to do this, we just ran a pretty straightforward thermal stress experiment on our coral Galaxea fascicularis with 3 different genotypes, which resulted in 6 different single-cell RNA-seq libraries comprising of 3 heated and 3 control to create this first cell atlas for this species. So here's the same cell atlas just split into the control and heated treatments. And in the middle here, we have a proportional bar chart, which shows the percentage of cells from each library for each cell type. So here we can see at the top that the immune cells, the algal hosting cells, neurons, and calcoblastic cell types are more enriched with the control atlas compared to the heat stress, suggesting that these cell types are actually decreasing under heat stress conditions. Contrastingly, we see that there's a different type of immune cell that is increased during heat stress, which is quite interesting, and we would have missed this with traditional bulk RNA-seq methods. When we look at what's actually happening at a gene expression level per cell type, this is basically just a bunch of volcano plots concatenated together with downregulation on the right-hand side and upregulation on the left-hand side. We see that gene expression is quite variable amongst all cell types. So again, we wouldn't be able to get this cell type resolution with traditional bulk RNA sequencing. But if we dive into just 2 specific cell types, like the algal hosting and calcoblastic, we see that the algal hosting cells are downregulating a lot of metabolic processes and ion transport, and upregulating a lot of innate immune processes and heat shock pathways as well. So it's worthy to note that these genes and pathways are really well described at a bulk transcriptomic level, so it's really nice to see that we can pinpoint which cells are actually expressing these genes and pathways. When we look at the calcioblastic cell types, we see that they're downregulating calcium ion transport and proliferation and cellular matrix organization, suggesting that calcification is being stunted during heat stress. And then we see that they're upregulating basically just genes trying to survive. So these cells are just in a really stressed state. So just to quickly summarize, I know that was quick, but through this method, we're able to understand which cell types are most affected by heat stress in corals, and then we can start to actually tease apart our bulk RNA-seq interpretations and pinpoint them to specific cell types. Thank you.